first start by thanking Wayne, Aldo, and Tanya for organizing this workshop. Thank you. While listening to Sean, I keep checking my slides. I thought this is the last talk, so I tried to make some points. A few of them are overlapping with Sean's presentation. Hopefully that would stimulate some discussions. So just to start by kind of mentioning that there's increasing recognition, right? No matter what you're studying, cognitive functions seem to be represented and processed using multiple brain regions, not just by one particular area at a time. So this is shown by a review on persistent activity during a delay period, underlying motor memory at the top, and an example of simultaneously putting from 6 different brain regions in a monkey performing a decision task, right? Suggesting that whatever you're looking at, you seem to see widespread signals. There's more data from mice studies to the point that there's a new paper at the bottom using the title something like "Not Everything, Not Everywhere, Not All at Once." Just indicating this kind of debate these days that on one hand we recognize that representations are distributed and widespread. On the other hand, how about the classic idea of functional specialization, right? As Sean mentioned in the example of detection task, there are brain regions where neurons seem to encode objectively physical stimulation, and then some other areas seem to be more engaged in subjective perception, right? Is there this kind of divide, right? So there's a divide. Where's the transition? How does the transition occur? Can we explain this modularity that's consistent and compatible with distributed representation? So that's the question I would like to address near the end of the talk. For computational neuroscientists, right, so the new challenge and exciting opportunity is to think about how to go from local circuits to large-scale brain systems. And we started in collaboration with Henry Kennedy, as Sean mentioned, who at that time, about 10, 12 years ago, published a very interesting database of inter-area connectivity that's directed and weighted. So suppose that we take this matrix and we want to build a dynamic model for a multi-regional cortex model. What do you do? How do you describe each local circuit, right? So that is a very wide field, right? Depending on the kind of questions you want to investigate, you have to kind of decide how to model each local area, right? We started with a very basic principle that also is I think consistent with Sean's talk. That is, let's start by assuming that local circuit obeys a canonical local circuit organization, right? You have this idea of repetition of copies of the same canonical local circuit across the whole cortical mental. But if that's the case, how do you explain functional modularity, right? If you think about mental processes, cognitive functions, there's clearly some specialization, right, of different parts of the brain. Well, usually people, like using graph theory analysis, would think about that the answer is input and output, right, which of course is true. So V1 gets very different input than PFC, right? However, the input and outputs are given by the weighted connectivity matrix, right? And what we're exploring is that on top of that, there are biological heterogeneities, right? And so this is the thing that I'd like to reiterate from what Sean said, right? So we know there's heterogeneity, that's not new. But what I guess it's interesting is that if you look at heterogeneities, they are not high dimensional. They're not just random heterogeneities across the cortex. You can actually do analysis and quantify some systematic changes along some low dimensional axes, which we call macroscopic gradients, right? One example Sean has already shown is the spine count per pyramidal cell, right? That seem to follow a very systematic increase along the cortical hierarchy. Another example from John Murray, looking at gene expressions. For some reason, I'm going to show later briefly that we think that recurrent excitation in higher areas should have slow dynamics mediated by NMDA receptors. And he found, for example, NR2B subunit encoding genes indeed show this increasing gradient across cortical hierarchy, right? Another example of inhibition is the ratio of input controlling somatostatin neurons divided by output controlling PV neurons shows pretty systematic change across cortical hierarchy. And finally, the last example to show is coming from Xiang's work, that DY receptor. This is now protein labeling density increases systematically along the cortical hierarchy, right? So, following these ideas, and this is the first example from Rishi Shukadari. When we say there's a chronic local circuit, we mean that the local circuit is described by the same mathematical equation, right? And the powers are very short, exactly the same for all cortical areas, right? Now, when we say there's a macroscopic gradient, for example, of excitation, quantified by sonic count, we introduce a gradient. There's a so there's this HI, right? For each area that might increase along the cortical hierarchy, right? So that's the gradient form. So Rishi actually started by simulating this model by driving each area with noise, mimicking resting state, right? And he found right away that the time scale over which the fluctuations occur is pretty different, right? Pretty fast in V1 and lower in PFC. You can quantify each by autocorrelation function. From there, you can extract the time constant. And you will find that, roughly speaking, the time constant during this kind of resting state seem to increase eventually along the cortical hierarchy, which we call hierarchy of time constants. This is an example where the effect curve is threshold linear. So if the input is above threshold, you are really looking at a linear system, right? For a linear system, you can think about time constants being given by each eigenmode, right? And so by analyzing eigenmodes, you could try to understand what, why, and how the hierarchy of time constants really emerge. So for that purpose, we borrowed a measure from physics called the inverse participation ratio. That roughly says that for each mode, indicated by a dot here, involves how many nodes? What's the fraction of nodes that are engaged in a particular temporal mode? Okay, the color here indicates the time constant, and each one corresponds to a particular mode. But that measure does not tell you, right, the nodes that are involved in a particular mode, whether they're physically together or far apart. So we introduced a new measure. I'm going to see the mathematical description, but just tell you that there's a second measure that tells you how close physically they are, right? Okay. So for example, if here we have 4 examples, right? So if you look at this one, both vectors are relatively low, okay, and it's blue, so it's kind of fast. We see that the nodes that are engaged in this mode are pretty widespread, right? But what's more interesting perhaps is compare those 2 examples. They have the similar kind of IPR, right? But one has a lower spatial localization measure than the other, and you see indeed that this one, is a lot more spatially localized than the other one. And interestingly, this one is slower, right, in orange, and that one is in blue. So you have some slow modes that are very spatially localized, and others maybe more widespread. So I think the time constant hierarchy is quite interesting functionally, right? That's what you want, because for early sensory areas like say retina or V1, you want the time constant to be very, very short in order to process information in the visual world that can change very fast, like watching a movie, right? On the other hand, I'm sure you heard about ramping activity underlying accumulation of information in decision making, say by drifted diffusion model. That assumes that the time constant is very long, right? And so somehow in this model, this hierarchy of time constant emerges, which is highly non-trivial, I think. John Murray looked at silver unit data from behavior monkeys and found some evidence that indeed, there's a gradual increase of intrinsic time constants along the cortical hierarchy in like a monkey. And there's a recent study showing that you have something like that in mice as well, right? Here's a unpublished study, a summary figure using ECOG recordings from marmoset cortex. That seems to also show this hierarchy of time constants. But I guess what's kind of surprising to me, because this is ECOG, right? Not really single units, that the range is pretty short, right? And it turns out that was found before in macaque as well. So this is a paper that shows time constant extracted from ECOG versus the time constant from a single unit from John Murray's analysis, right? You do see that the two correlate very well, but the range seems to be very short. Nevertheless, I think this means that you could, in principle, look at this, time constant hierarchy if you use ECOG measurements, perhaps in humans pretty well. Now, the other maybe useful comment here, following previous talks, is that the gradient of excitation, heterogeneity, actually has a big impact on understanding resting state functional connectivity, right? When you just think about a graph, you kind of implicitly assume that nodes are identical. They're all the same, right? But what we found is that, what we suggest is that because areas are actually heterogeneous, because of the gradients, the relationship between functional connectivity and structural connectivity, which we know in the model, right, measured by Kennedy, could be, better understood if you take into consideration of macroscopic gradient. And that idea has been tested by John Murray, and indeed, their funding is in support of this idea. Now, let me move on to talk about working memory. So here, it's the same connectivity data that we use from Henry Kennedy, but now for each local circuit, for the sake of looking at working memory, we're going to use a highly nonlinear local circuit model. I think Sean mentioned it. I'll just talk a little bit more about that. In each area in this simple setting, you have two selective neuron groups, right, for, say, stimulus A versus B. That way, you can talk about stimulus selectivity in a working memory task. You can also look at, stimulus versus the disruptor, for example, right, in working memory tasks. And the two compete through shared inhibition, right? So again, each local circuit is described by the same mathematical model. And we actually borrowed this from a previous study, which we developed that can account for certain salient physiological observations about working memory and decision making. Namely, when you try to make a hard decision, for example, try to discriminate the motion direction in a random dot pattern when the motion strength is very weak. You see this duality, right? On one hand, there's this COSYNE ramping activity that you may be familiar with as a mechanism for integration of information. On the other hand, there's this winner-take-all competition leading to a choice, right? A winner. And that's the same mechanism that can maintain working memory trace in form of persistent activity. So we're going to use exactly the same model like this, but as you, I'm sure, know that this kind of behavior happens only when the strength of the kind of excitation is strong enough, right? So for this kind of local circuit model, there's a bifurcation diagram, right? So along as function of the strength of the kind of excitation, you have this bifurcation, right? Below that point, you don't have anything really interesting except the resting state, except responses to a sensory input, but there's no perceptivity, there's no ramping underlying decision making. But when you're above this threshold level for JEE, there's this emergence of working memory and decision making capability, right? So now imagine that we use this, and JEE is going to change from area to area along the coding hierarchy, right? Say in the form of a gradient. So basically, along the hierarchy, each area has a position. There's a V1 is here, V2 is here, V4 is here, etc. Each one would have a JEE value, right? The one at the top would have value that we call Jmax. And by the way, this is work by MRDS, who's here. And here explore the different scenarios. Where's Jmax, right? You'd say, well, I see this activity, I see working memory related signals in certain areas like LIP and prefrontal cortex. Does that mean that each of these areas, the local E to E coupling strength is above the threshold? We don't really know, because you do recordings from behavior animals with intact interactive system, right? So that's why when I look at the situation when Jmax is below the threshold, in which case none of the isolated areas is capable of working memory and persistent activity. And what you see in the interactive large-scale system has to depend on connection loops between areas, right? But of course, in the model, you can also explore other scenarios when Jmax is near the threshold or above the threshold, right? So what's shown here next is an example where Jmax is below the threshold. So what you see is an emergent phenomena of the multi-regional system, right? This is a simulated delayed visual response task. You see V1 respond vigorously to the input, but it gets away when the input is gone. Similarly for MT, but LIP and some frontal areas show persistent activity that outlasts the stimulus, right? So one way to look at the same data is to look at activity map. During stimulation, you see the activity is mostly in the posterior part of the cortex, whereas during the delay period, you see this is distributed working memory representation, right, involving multiple brain regions, but not V1, for example, right? So this is where I can propose perhaps a definition of modularity, right? So by modularity, we don't mean that one function is done by one isolated area. We mean that it's dependent on a dedicated subset of areas, which may or may not be physically together. All right. And here I'm giving you actually a few more examples again from Boy's work. So each of the 6 examples represents a self-sustained attractive state, right? Some can be rather localized, like this one. Right? Some are more distributed. This is what I meant, right? So this is a subnetwork involving a subset of areas representing internal state, which can be used for a certain cognitive function potentially. All right. So the same network actually can display many self-sustaining attractive states, each one engaging a subset of areas. Xiao mentioned, I'm going to flash out this mouse modeling paper that just came out. What's kind of interesting is that it turns out that in mice, there's no evidence of gradient of spine counts. This is shown by a very interesting study where they replicated the monkey data, spine count data, for monkey V1 in pink and the monkey PFC in red. Right? You see big difference. That the same lab using the same method applied to mice, they did not see any difference between V1 and frontal areas. So we actually don't have a clear evidence that there is a gradient of something that indicates that excitation strength increases along the cortical hierarchy next. Instead, Xing used the gradient of inhibition. So PV neurons are the ones that control the output of younger cells, and they are, of course, critical for controlling excitability, right, as we know. That turns out to be very different across different areas. So the idea is that maybe the areas that have lower peak density may be more prone to show persistent activity, right, in a large-scale system. And indeed, that's what we found. So for the sake of saving time, I'm going to skip some details of this paper. I think it's a mice model, so we can potentially really relate such a model better with a lot of experimental measurements. But I just want to end with one point. So the same data, both in monkey and mice, if I do the delay period, we look at the working memory representation, right? If I just plot the fine rate of persistent activity along a function of hierarchy, you see something like this, right? Those areas that are engaged in working memory are separated from those that are not by a gap in the fine rate. That looks a bit like a bifurcation diagram of the local circuit, right? So we wonder, mathematically, does this emergence of modularity correspond to some new form of bifurcation? I just want to mention that this is highly non-trivial for several reasons. Number one, as I mentioned, right, if you disconnect areas, everything goes away, right? This is going to disappear. And number two, the connectivity is very dense at the level of area connectivity. So a lot of long-range connections, and yet there's this kind of a spatially localized transition, right, that occurs. And number three, this is true for each of the attractor states. If you have many attractor states, this phenomenon is applying to each of them, right? Okay, so this is a highly non-trivial. So I'm going to skip some details about a very interesting, I think, mathematical analysis of this phenomena using a generative model. Because if you want to look at a transition, like a phase transition, for example, you want to get very close to the transition point, right? In order to do that, we use this generative model where you can generate a super cortex with any number of areas you want. It can be 1,000 areas, it can be 2,000 areas, with the same statistics of connectivity as in the monkey cortex. So we did that, and we replicated the same kind of phenomena in blue during the delay period, right? You see this, firing pattern. And in red is reanalyze the time scale fluctuations for each area during the delay period. Now you don't see this roughly increasing gradient of time constants, right? You see this inverted V-shaped time constant. There seems to be some kind of divergence of time constants right near the gap of the fine rate, right? That is a phenomenon in physics called critical slowing down, indicator of phase transition. So we went back to the connectome-based model. We basically found the same thing. So you say, well, how can that happen? You just told me that the time constant should go up monotonically. Well, the answer is that the system is nonlinear. Time constants are really defined for a linear system or for a given internal state, right? You look at fluctuations around an internal state. So indeed, the same model shows this monotonic hierarchy of time constant at rest, but shows this inverted V-shaped pattern of time constants during the delay period, right? And similarly for the mice model. So that's a very specific prediction. We'd like to test that. It's totally testable, I think, although technically not so easy by looking at time constants both during the resting state and delay period. So let me just end maybe with one take-home message. Namely, this is just starting, right? We know this field is really just getting started. And the question is how far we can go to assume a canonical local circuit, which is a very powerful idea, and adding macroscopic gradients of biological properties and look at emergence of modularity, perhaps in the form of bifurcation in space. Thank you.